Welcome back to Day 3 of NCERT Biology 365. Today, we enter Section 8.4 to dissect prokaryotic cells. Prokaryotes multiply much faster than eukaryotes, and their genetic material is unique. NCERT points out that in addition to the genomic DNA, many bacteria have small circular DNA molecules outside it, called plasmids. Highlight this. Plasmid DNA confers unique phenotypic characters, such as resistance to antibiotics. This is a prime target for statements on bacterial genetics.
NCERT states that most prokaryotes have a chemically complex cell envelope. This envelope consists of a tightly bound three-layered structure: the outermost glycocalyx, the middle cell wall, and the innermost plasma membrane. Make a note of this line. Although each layer of the envelope performs a distinct function, they act together as a single protective unit. Under exam pressure, a statement might claim these layers act independently. Remember, they function as a single unit.
How do we classify bacteria based on this envelope? Christian Graham developed a staining method that splits bacteria into two groups. This classification is based on two factors: differences in their cell envelopes and their response to the Gram stain. Those that take up and retain the stain are Gram-positive, and those that do not are Gram-negative. Note both criteria for your statement-based questions. The outermost layer, the glycocalyx, is highly variable. NCERT warns us that it differs in both composition and thickness among different bacteria. In some, it is a loose sheath called the slime layer. In others, it is a thick and tough layer called the capsule. Examiners love to swap these definitions. Remember, slime is loose, capsule is thick and tough. Let's look at the remaining layers. The middle cell wall determines the shape of the cell and provides strong structural support to prevent the bacterium from bursting or collapsing due to osmotic pressure. NCERT notes that this membrane is structurally similar to that of eukaryotes. Mark this comparison for structural questions. We have covered the prokaryotic cell envelope. Now, head down to the description, click the link, and take the Day 3 CBT quiz. Tomorrow, on Day 4, we will study the inner compartments of prokaryotes: ribosomes, polysomes, and inclusion bodies. Click to subscribe and watch the podcast episode, and keep your streak going.